afternoon, everybody. Welcome back to our last virology lecture for spring 2020. We started this course way back in January in lecture number 1 with a brief mention of a new coronavirus that was spreading globally. And I thought it would be fitting to start our last lecture with an update on where we are. This map is very different from it was back in January. Most of the cases were in China. Now we have over 3.5 million total global cases with 250,000 deaths. And look at the country that has the most cases. The U.S. with 1.2 million, followed by Spain, Italy, UK, France, Germany, Russia, Turkey, Brazil, Iran, and then China is way down. They did a great job at containing this. And we have Italy with the most deaths. If we take a look at the daily cases on this slide, when we started this course, we were back here. Anyway, way back here, and we watched this curve grow, and then it got out of China. And if you look at it now, it seems to have plateaued at about 70, 80,000 cases a day. That's a lot of cases, of course. So each bar is cases per day. And in certain countries, it is declining. Let's take a look at the U.S. here and go to here. That's the U.S. Yeah, there are peaks and valleys, but it seems to be trending lower. If you look at cities, New York City is going lower. And so everyone's talking about getting back. However, it's a complicated issue. We don't have time to deal with it today, but I'll tell you how to keep listening at the end to follow what I'm saying about this. Today I want to talk about therapeutic viruses, kind of a culmination of this course. Everything you've learned, we're going to put together here and really see how we can manipulate viruses to help us out. What do I mean by that? These are the 4 different areas I want to cover today, which I call therapeutic viruses. Using viruses for good, at least our design, because many of them are good, as you have guessed throughout this course. First, we're going to talk briefly about phage therapy for bacterial infections. Then we'll talk about gene therapy, delivering a gene to patients who lack the gene or have defective versions of it. And we can use viruses to deliver antigens, for example, in viral vaccines. We've mentioned this already. And finally, using viruses to kill tumors, viral oncotherapy. What makes all this possible is the ability to manipulate viral genomes using infectious viral DNAs. This is a key for vector development. We talked about this way back in lecture 3, I think, genomes and genetics. For every virus, you can make a DNA copy and modify it. For poliovirus, you can make a DNA copy of the RNA. You can modify the DNA. In the case of vectors, modify its tropism, add genes to it that you need, add antigens, recover the viruses, and then use those in patients. So this is an essential technology. Without this, none of this would be possible. And this comes from basic science, basic molecular biology research. First, a few words on phage therapy. You may remember that bacteriophages were discovered in 1915. There were 2 discoverers. One of them, Derel, continued to work on them to treat bacterial infections. He really thought they had potential. He co-founded in 1923 the Eliava Institute, which is in Georgia, and is active to this day, making phages to treat bacterial infections. In fact, during World War II, this institute produced phages for antibacterial therapy. However, the introduction of antimicrobials in the 1930s and later on really put a lid on the development of phage therapy because the antimicrobials were so much easier to develop. And they, of course, took care of many infections. Nevertheless, subsequently, we know emergence of widespread resistance to these drugs has made us look elsewhere to treat bacterial infections. And guess what? Right in front of our eyes, we have phage therapy. And the Eliava Institute's been working on it ever since, and many companies are now interested in it. Just give you a little update on where we are. Of course, the principle is you use a lytic bacteriophage to kill a host. So phages can be lytic or lysogenic. Lysogenic meaning the genome is integrated and the phage does not kill the cell. So you don't want that. You want a lytic phage, and you want to kill specific bacteria that are the problem. You don't want to kill all of your beneficial microbiome, so you have to tailor these. You have to identify the pathogenic bacterium, and you have to find phages that will kill it. You have to do all of this before the treatment. So this is not trivial, and this is not our usual interpretation of what a drug is. So it challenges the FDA, for example, and new approaches are going to have to be developed. Another variation of phage therapy is to actually use phage lysins. These are the enzymes that help to pop open the bacteria for lysis. And the way they work is they digest the peptidoglycan, for example, and then the membrane will burst because of the pressure from below. And these have been used for surface decontamination, especially in food preparation areas where surface bacterial contamination is an issue. You can use these lysins and even phages, in fact, to treat it. And these are two licensed phages. This one is Phage. It's approved by the EPA to treat bacterial tomato canker. So in the fields, the tomatoes are having this bacterial infection. You can spray this phage on them. It works. It's licensed. It's approved by the EPA. And then List Shield. This is a bacteriophage that kills Listeria, common food contaminant, common contaminant of meat and poultry. And it's approved by the FDA for treating the meat and poultry, not treating people, just treating the food. And it actually was the first phage product to get the designation of generally regarded as safe. And there have been some spectacular recent clinical successes with phage therapy. I just want to highlight here. Here is one phage treatment of an aortic graft. This patient had gotten an aortic graft. It became infected with Pseudomonas. They weren't able to do anything, but they squirted the phage onto it, and it cured this infection. And Paul Turner's one of the authors here is from Yale University. I interviewed him on TWEVO 44. Really interesting stuff. Then we have another paper here. Engineered bacteriophages for treatment of a patient with a disseminated drug-resistant Mycobacterium abscessus. This was a young individual who had this disseminated infection, wasn't responding to antimicrobials. They developed phages specifically for this bacterium, and these phages had come from a high school project where high school students go out in the soil and they recover dirt and look for phages that infect mycobacteria, and they use some of these, actually engineered these further to be specific. So another key here is to engineer the bacteriophages in ways that will be beneficial. And finally, development and use of personalized bacteriophage therapeutic cocktails to treat a patient with a disseminated resistant Acinetobacter infection. And this was a case where an American developed Acinetobacter overseas. It was resistant to all antimicrobials. And then a group at the Navy and Texas A&M came up with a phage to treat this, and they gave it to him intravenously. And I think after 10 days, he woke up from his coma. And so that's one of the papers describing this. And we went to Texas A&M 2 years ago, and we talked with some of the principals who did that. So I highly recommend listening to these to really get an idea from the people who did the work. That's all we're going to say. I suspect in future years we'll say more in this lecture, but right now it's a growing field with lots of potential. I want to talk about using viruses as vectors, and I'm going to go through a few vectors and their uses. First, adenovirus is a very popular vector. This is a large DNA-containing virus with a 40 kb-ish double-stranded DNA genome, which we know a lot about this virus, its tropism, what genes are essential and non-essential, so we can modify it to put in genes. These vectors have a lot of advantages. This is a big one, efficiently infecting post-mitotic cells. That means cells that have stopped dividing. Many viruses won't infect those, so you have to find viruses that will, and this is one that will. Very fast onset of gene expression. The genome stays episomal, so there's no insertional mutagenesis. It's big capacity. It's easy to make pure concentrated preps. There are lots of human serotypes. There are also animal serotypes. You may just have heard of a chimpanzee adenovirus being used for a SARS-CoV-2 vaccine. That means that if you run into immunity in the population, you can switch serotypes. And this, of course, is one of the drawbacks, that you inject and then the person will make antibodies to the vector. A little bit of history on these vectors. First generation, it took the genome and simply deleted the E1 and the E3 regions. Now, you may remember E1. You should. You've heard about this an awful lot in this course. This encodes the T antigens that abrogate, of course, RB and P53. That's down here. You can complement this function from cells. So the vector itself, you require these to reproduce, of course, because they kick the cell into dividing, but you can provide this in trans in a cell line. And E3 encodes immunomodulatory proteins, which, by the way, are not needed for reproduction in cell culture. So that's first generation. Second, they learned what other genes they could take out. So besides E1 and E3, E2 and E4 to make more space for the transgene. And that brings us to what we call third generation or gutless vectors. All the genes are deleted. Only thing left are the inverted terminal repeats and the psi, the packaging sequences. So here's a diagram of that vector. You have the two inverted terminal repeats required for DNA reproduction, of course. Then you have a very short piece of DNA, 500 base pairs or so, which contains a promoter, and then packaging sequences. So this can be packaged in the particle, and then you put your transgene in here. So most of the genome is removed. That's why we call them gutless. You have to give this vector capsid proteins because it doesn't have its own. So you introduce the vector DNA, which has, again, the ITR's packaging sequences, promoter and transgene. That's what we call vector DNA. You introduce that in cells that have been infected with a helper virus that will provide the capsids. But you don't want the helper DNA to be packaged, right? That would be no good because then you would have wild-type virus present. So what you do is, this is first of all deleted for E1. So you have to use cells producing E1 to propagate the virus. And then the helper, okay, so the helper is deleted for E1. The helper also has LOX-P sites flanking the packaging sequences. What are LOX-P sites? These are substrates for a nuclease called CRE. And when CRE sees these LOX-P sites, it will cut them and remove the packaging sequences. And so what you do is you infect with a helper adenovirus with this genome configuration, delta E1, LOX-P, in cells that make both CRE and E1. So E1 allows this to reproduce. The CRE will cut out the packaging sequences of the helper, but the helper will still make capsid proteins to package the vector DNA. So you have now particles with only your vector DNA. These particles will deliver the transgene to cells, but they won't make new particles. So that makes them safer. That's adenovirus. We also use adeno-associated virus a lot. Remember, these are the smallish viruses with single-stranded DNA genomes, which don't encode many proteins, basically a rep orf and a capsid orf. And you make vectors essentially by deleting most of those sequences. You delete rep and you delete cap, and you make a transgene vector with the ITR as a promoter, and you put your transgene in there. Now, to make vectors, virus particles, you have to transfect that vector plasmid into cells along with a plasmid that produces rep and cap so that this will replicate and make capsid proteins and get packaged. But because this is a helper-dependent virus, Adenovirus-associated virus, right? You have to add adenovirus helper functions, which include E1A and B, E2, E4, and the VA RNA gene. So all those things on plasmids, vector, rep-cap plasmid, out comes recombinant AAV that has this DNA only in it, ITRs with your transgene. And these are beneficial. They give you long-term expression, and you get multiple serotypes. The long-term expression is really interesting. Now, AAV genomes, they can integrate into the host cell in the absence of a helper, but that integration requires the rep proteins that we don't have in these vectors. So these vectors are not going to integrate. Nevertheless, they do persist, and that's because the vectors recombine to form circles. You can make monomers or concatemers. This was totally unexpected that in the absence of rep, these things would make circles. And the circles persist for reasons that we don't understand. So the way this works is the genomes enter the nucleus, double-stranded DNA forms, either by synthesis of the complementary strand, and that requires only host proteins, no viral proteins. Or sometimes, these particles package both plus and minus strands, they anneal to make a double strand. And these have ITRs, of course, and recombination between different of these molecules, because you get multiple per cell introduced, give you the circle monomers or concatemers. And those are what persist. And that's why you can have mice, for example, and people too, that have these for years. Great feature of a vector. All right, now we have retrovirus vectors. And these are based on retroviruses with simple genomes with gag pollen envelope, or retroviruses with complex genomes like lentiviruses, HIV-1 based vectors, where you have gag pollen envelope and lots of accessory proteins. And so what you do is you have a whole bunch of different plasmids here. You have a packaging plasmid. The packaging plasmid gives you everything you need to make a particle. And so it's a plasmid with a foreign promoter, and you have gag and pol on them. And so these plasmids don't have any packaging sequence, so that's not packaged as particles. And then you have an envelope protein. This gives you the glycoprotein. It's the same, so have it under each of these vectors just for completion, but these are basically similar. And these envelopes can come from other viruses like VSV, and we call that pseudotyping. So you get a broad host range. And then you have transfer plasmids where you would put your gene of interest in. You would have here in red your gene of interest, and you have LTRs and a promoter. It could be a different promoter. It doesn't have to be the LTR-based promoter. And this will deliver your gene. And I'll show you how to get these all in a particle in a minute. We now have what are called self-inactivating transfer plasmids, or SIN plasmids. These have minimal LTR sequences. A large part of the U3 region is deleted in these vectors, and so that after reverse transcription, you can't reconstitute a full-length LTR. And so these will not have promoters, for example, and enhancers that can activate downstream genes. So you may have to go back to reverse transcription to figure that out, but we've made these not possible to make full-length LTRs, and that makes them self-inactivating and safer. So the way you would make these vectors is you would take your capsid plasmid with gag pol and your envelope plasmid, you put them in cells and out come viruses without genomes, empty. If you want to put a genome in, which of course what you want to do, you include a transgene plasmid made as we just showed before, and this gives you a retrovirus containing a foreign gene. These have lots of advantages, these retrovirus vectors. And again, they're based on lentiviruses or other retroviruses. HIV is unusual because it can infect non-dividing cells, whereas the other retroviruses typically cannot. You get long-term expression, of course, because the provirus is integrated up to 8 kb inserts. However, you do have the possibility of insertional mutagenesis when they integrate near a gene, and so that for that reason we have made these inactivated LTR SIN vectors, or even ones that can't integrate. But in that case, of course, you're losing a great advantage. But you can make them safe without doing that. And we can pseudotype them with other glycoproteins like VSVG, which gives them a broad host range. We use poxviruses as vectors, and one of them is called modified vaccinia virus Ankara. This was a virus originally made as an alternative to the smallpox vaccine. So the smallpox vaccine is based on vaccinia virus or related virus, cowpox, horsepox virus. And this is a modification of that vaccine. And this has actually been stockpiled by the U.S. in case of bioterrorism. There's enough to vaccinate every American with this vaccine. Look at that. We're looking ahead for smallpox. We couldn't look ahead for coronaviruses. This is a replication deficient vector. It was passaged extensively in chicken cells, and they selected a virus with an assembly block. So you can grow up stalks in bird cells, but not in mammalian cells. It will go in, but not replicate. And so it's a great vector. This can be worked on under BSL-1 conditions, very safe, has a large capacity, And so we can put big genes in here. We know how to make lots of it and purify it. And this is also another version of canary poxvirus has been used in the same way. These viruses, of course, have been used a lot in biomedical research. So we know a lot about them. And this particular modified Ankara is quite safe. And many vaccines have been developed using this platform. For example, influenza H5N1 vaccines, many HIV vaccines have been tested here, West Nile, SARS, MERS, and the MERS vaccine using this platform is being used for camels, so that's a One Health vaccine approach. And of course, this is being used to make one of the many SARS-CoV-2 vaccine candidates. So this is how you would insert a gene into modified vaccinia virus Ankara. You would have a transgene in a shuttle vector. This is a plasmid where you have in red the gene you want to put into the vaccinia virus, and then flanking it are vaccinia virus sequences that are recombination sequences. So basically you transfect that into cells, you infect with MVA, and these purple sequences will mediate recombination with the genome. And so out will come viruses where your transgene has been introduced by recombination, and you just screen for them. Another virus that's been used as a vector is vesicular stomatitis virus. We've encountered this. Its use as a vector for making vaccines, the Ebola virus vaccine. So this is a negative strand RNA virus, which encodes for a number of messenger RNAs, including all the proteins you need to make a virus, and one of them is the glycoprotein. And typically what you do is you replace the glycoprotein gene with a transgene. That attenuates the virus so it doesn't cause disease in people. And the way you do this is you have a 4-plasmid transfection. You have the plasmid encoding the whole RNA genome with your transgene in it, and then N, P, and L plasmids. That gets it to replicate, and then you put that in cells. They have T7 RNA polymerase promoters to drive the synthesis of RNA from each plasmid, and so you co-infect with vaccinia virus that expresses the T7 RNA polymerase to drive RNA synthesis from these plasmids, and out come infectious virus with your glycoprotein of choice. We'll talk about some examples of its use. Flaviviruses have been used for vaccine vectors. We talked about how the yellow fever vaccine virus was cloned as a DNA copy, and then the structural proteins were replaced with those of dengue virus, and we now have a dengue virus vaccine, Dengvaxia, where for each of the 4 serotypes, we've substituted these with the dengue virus proteins. And because this is derived from yellow fever, it's already attenuated. And so it's just a matter of replacing these proteins and testing them in people. And so the way this works is you have a plasmid encoding this vector. You make RNA in vitro, and then you transfect the RNA into cells to get out your recombinant viruses. Now, an interesting alternative to this is the use of flavivirus vectors based on flaviviruses that infect insects. These are insect-specific flaviviruses, and one that's been used is called Binjari, Binjari. These do not replicate in vertebrate cells. But if you put foreign genes in them and introduce them into vertebrate cells, you can get an immune response. So, for example, these structural protein genes have been in Binjari. So imagine this is Binjari virus, an insect-specific flavivirus. They can be exchanged with dengue, Zika virus, West Nile, yellow fever virus, Japanese encephalitis virus. These are all flaviviruses. They replicate really well in insect cells. So you make stocks from insect cells, and then you can immunize animals. And in one experiment, mice were immunized with Binjari with the Zika virus structural proteins, and this protects mice from disease after Zika virus challenge. So this is a very interesting approach, using insect-specific flaviviruses instead of human flaviviruses. There's another approach employing alphaviruses. There's some alphaviruses that only infect insects. One of them is called Eilat virus, E-I-L-A-T. And so what you can do here's the alphavirus genome. It's a plus strand RNA that encodes 2 open reading frames, the replication proteins, and the structural proteins from a subgenomic mRNA. You can substitute these structural proteins with those of, say, chikungunya virus, which has been done. So islat virus, imagine this is islat virus. You substitute the structural proteins from chikungunya. You make a recombinant virus in mosquito cells. Those viruses will not productively infect mammalian cells, but if you immunized mice with them, they make antibodies that protect them from a lethal challenge. They've also done experiments in non-human primates where a single dose has protected against chikungunya infection. So in other words, we have two insect-specific viruses, one a Flavi, one a alpha, where you can assemble the vectors in insect cells and then use them in mammalian cells. They're sort of like inactivated viruses because they don't replicate, but they do get into the cell and they make immune responses. So I like these as potential new vaccines, and some of these are being tested for SARS-CoV-2, of course. And finally, Newcastle disease virus. I don't know if we've mentioned this. This is a paramyxovirus. Happens to be a disease-causing virus of chickens. Really important economically. And vaccines have been made for years to try and protect chickens. And what they did here was to develop this basically as a vaccine strain. And now it's being used for other antigens. And so it's a negative strand genome, very much like VSV encoding a subgenomic mRNAs. And typically you put your transgene in between the P and the M genes. And the strategy is similar to VSV for recovering recombinant viruses. 4 plasmids, NPL, transgene RNA plasmid with T7 promoters. You infect with a T7 RNA polymerase producing vaccinia virus, and out come your Newcastle disease virus with your gene inserted into it. And in fact, so these have been used to immunize chickens just as they are with no modification, but they have also been used to protect chickens against avian influenza viruses. So you can put in a transgene here, say a hemagglutinin from H5N1 avian influenza virus, and now you immunize with that and you get protection against both Newcastle disease and H5N1 avian influenza virus. You get 2 vaccines with 1 shot. Pretty good. So some of these that we've talked about are licensed as vaccines. Of course, Ervebo is VSV with the glycoprotein from Zaire Ebola virus. This is licensed for protection against Ebola virus disease. Dengvaxia, of course, is dengue virus and a yellow fever vaccine vector. Yellow fever vaccine vector has also been used for, as I said, Japanese encephalitis virus in humans. West Nile vaccine in horses is available to protect your precious horse from West Nile. And then, as I said, the Newcastle disease virus vector has been developed to carry H5 avian influenza virus to protect chickens. So here are some experimental human vaccines that use virus vectors. There have been a variety of HIV vaccine candidates. I shouldn't say human vaccines, actually. Vaccine candidates. It's not the same thing. These aren't licensed. So one has been used extensively for HIV. These have all failed. All the HIV vaccines have failed, including this one. Adenovirus carrying Ebola virus glycoprotein will probably be licensed at some point. We have MVA modified vaccinia Ankara carrying H5 of avian influenza virus. The RV144 AIDS trial that we talked about last time is a prime boost, which includes modified vaccinia Ankara carrying the glycoprotein and a canary pox with the glycoprotein as well. We have the MVA MERS-CoV in camels. AAV vectors also being tried for many viruses, herpes simplex, human papilloma, HIV, SARS coronavirus. There's a experimental VSV MERS-CoV. And basically all these platforms are being tested for SARS-CoV-2. Let's move on to gene therapy. Typically we do gene therapy using viral vectors for monogenic diseases. That is a disease caused by a mutation in one gene. And there are diseases caused by multiple mutations in multiple genes, those are more difficult. So we're trying the easy ones first just to get the technology to work. So you have a mutation in a gene and it has an inheritance pattern which you can observe, which can be dominant or recessive, of course. These are quite common. There are over 6,000 monogenic diseases of humans, and they occur in 1 out of 200 live births. It's very frequent. So these are amenable to viral gene therapy, and at the moment there are over 1,800 clinical trials for these in progress, probably more. And here is a chart which shows you some of these just to give you an idea of the disease, the defect, and the incidence, and how we're treating it with what vector. So I've explained all these vectors to you now. You see AAV and retroviruses are big ones. SCID, immunodeficiency, bubble babies. They have to live in a very sheltered environment, otherwise they have no immune system. Some of them have an adenosine deaminase deficiency. Some of them have a defect in the common cytokine receptor gamma chain. These are the frequencies. I will tell you some results from that today. Lipoprotein lipase, hemophilia, hemoglobinopathies, and thalassemias, alpha-1 antitrypsin, and then we have blindness, retinal degenerative diseases we'll talk about today, adrenoleukodystrophy, and Wiskott-Aldrich. And these are just a sampling, so you get an idea of the defects in the frequency. So we're going to go through a few of these, and I'll give you some idea of what's been going on. And these are simply the number of clinical trials for gene therapy from 1989 to 2018. Number, y-axis, year on x-axis. They're going up, obviously. Look at this, very high now, and this is going to increase. And viruses are going to end up curing a lot of issues of humans. They cause them, I understand. We could prevent that, but they're going to cure a lot of them as well. And I have this slide to show you the indications that are being addressed. This is a bit old. This is 2017, and I'm sure the numbers are all different and higher, but just to give you an idea, on the left are the indications, on the right are the vectors. So, cancer is the biggest. Monogenic diseases, infectious diseases, that would be vaccines, and then a variety of other diseases as well. And then let's see where the most popular vectors: adenoviruses, retroviruses, adeno-associated viruses. Now this is much higher than shown on this slide. These are much higher. But then you also can give naked DNA. You can do liposuction with a facilitator, putting naked nucleic acids into people as well. All right, so that gives you a little bit of an idea. And in general, the way you deliver these, there are two ways that you would deliver a virus vector to patients. You have direct delivery and you have cell-based delivery. All right, so you make your virus, you put your therapeutic gene in it, you make virus particles and you inject them. Now this has limited utility because you would typically inject these intravenously, and 90% of them are going to be cleared in the liver. So if you have a liver issue and you want to infect liver cells, that's great. But it's really hard to target other tissues. And this is an issue for cancers, of course. Sometimes you can't inject directly into the cancer. You have to give the virus IV. So we're trying to figure out ways so that the virus is not bound up in the liver. But another approach is cell-based, where you either take cells out of the patient. You could take stem cells. You can purify these from the blood. You can put them in culture. Then you can take your virus with the gene of interest, infect the cells, make sure they're infected, check protein production, expand them, and then you reintroduce them. And because they're stem cells, they're going to populate the human and make the progeny of various sorts. So that's good that they have the gene. And you can do this with other cell types as well. Or you can take a variety of other cells. If you don't have stem cells, you can take embryonic stem cells and differentiate them into stem cells. And of course, you can also take somatic cells from the patient and make them into stem cells, introduce your virus, and then put them back in. All right, so those are the two general approaches. You either put the virus in or you put the virus in a cell of some kind and put the cell back. So let's go through some of the history. Early human viral gene therapy. One of the first trials, 1993, for cystic fibrosis. This is a disease where you have mutations in the cystic fibrosis transmembrane conductance regulator that predisposes to serious respiratory infections. So this was a male who was homozygous for a deletion or mutation in this gene, and he was given an adenovirus, E1, E3 deleted, so an early version, containing the wild-type gene, 2 times 10 to the 8th PFU, and this was administered in the airway. You can see them doing it here, actually. You have the doc. He's got a tube down into the trachea, and they're just spraying in the adenovirus. Then these images are pre- and post-therapy. They take some cells out and look, and this is stained for adenovirus proteins, or probably the CFTR protein, I would guess. And here on the right, they're measuring the exogenous versus the endogenous CFTR. And so you can see after the first administration of adenovirus, you get a nice increase in the transgene delivered by the virus. And then it goes down, and then you give another one, and it goes up again. But then by the third time, it doesn't go up anymore because the antibodies have kicked in, and that particular serotype doesn't work. So this is something we learned from this and modified subsequent trials. But that was an early one that said it might work, although a lot of these patients weren't fixed by this rather transient administration. Then we have the story of a setback. Jesse Gelsinger, 18-year-old, first person to die in a gene therapy trial. And this was a trial to treat ornithine transcarbamylase deficiency. This is an X-linked disease that leads to high levels of ammonia and glutamate in the blood. And this is not good because you have declining cognitive ability and premature death. So in the late 90s, I'll tell you the story about this. This was a phase 1 trial to evaluate the safety of an adenovirus vector with the normal OTC gene. And this was done at UPenn. It's a dose escalation trial. They had 18 patients who got a single dose delivered by the hepatic artery and 10 to the 9th to 10 to the 11th virus particles. It's a dose escalation study. And that highest dose, 10 to the 11th, 6 times 10 to the 11th particles per kilogram. It's a lot of particles. That was 17-fold lower than the lowest toxic doses that were determined in macaques. And the first 17 patients had a variety of adverse reactions like flu-like symptoms, which is inflammatory response, transient liver toxicity. They all made neutralizing antibodies, but none of them had their OTC deficiency corrected, unfortunately. And that's what had been found previously. Now, the 18th patient was Jesse. He got the highest vector dose. So the 17th and the 18th patients got the highest vector dose. Both of them developed liver toxicity, thrombocytopenia, and high blood concentrations of pro-inflammatory cytokines like IL-6. This is also an issue for SARS-CoV-2 patients. Unfortunately for Jesse, these reactions did not dissipate, but he went into intravascular coagulation, multiple organ failure. He died 98 hours after receiving the vector. And we don't know entirely what was the difference between him and the 17th patient, but later on they found pre-existing anti-adenovirus antibodies in his blood. This was blood taken before the trial. That may have enhanced transduction of dendritic cells, kind of like antibody-dependent enhancement. So that may have contributed to it. But this trial was stopped, of course. The FDA actually suspended all other trials at UPenn, and the FDA and the NIH reviewed this and many other gene therapy trials, especially ones involving adenoviruses. And at UPenn, they found various irregularities, including potential conflicts of interest in the fact that the toxicities found in macaques had not been reported at the time at which the trial was initiated in humans. So this whole program of gene therapy was revamped. From 1989 to 99, there had been a steady increase in the number of clinical trials. Because of this, the whole field stopped and it didn't recover for a decade. So that's what can happen if you don't do things properly, which was what happened at UPenn. Another disease, X-linked severe combined immune deficiency. These people have a defect in T, B, and NK cells, very susceptible to infections. They have mutations in the IL-2 receptor gamma chain. Okay, and that gene is shown here, and all these circles and triangles are mutations that have been identified in various patients of different sorts, different parts of the protein transmembrane, etc. Anyway, so there were 2 trials done in London and Paris giving infants, you have to treat this early because otherwise you have a horrible life, with a retrovirus with a normal gene. So what they did is they took out their bone marrow precursor cells, they infected them. Whenever you see transduced, that means they infected with a vector with a gene, transduction, and then they put them back into the patients. 4 of these 9 infants in Paris and 1 in London developed leukemia 3 to 6 years after treatment. The treatment actually worked, but then they developed leukemia. So all these trials, 27 ongoing trials with retroviral vectors, were halted until they could figure out what went on. And here's what happened. The vector integrated next to a cellular gene involved in uncontrolled cellular growth, this gene called LMO2. And these are 2 insertions of the vector that was used for gene therapy. And so this gene, LMO2, is actually known to be expressed abnormally in a form of childhood acute lymphoblastic leukemia. And we know that the insertion had to do with their cancers because their cancers were monoclonal, deriving from a single cell. Now, the patients who developed these cancers, so their disease was originally successfully treated. They developed cancers, and those cancers were actually treated with chemotherapy, and they survived. So this showed that gene therapy can work, and because of this, they modified retroviral vectors so they can integrate next to an oncogene. But you can see here, the reason why these integrations turn on LMO2 is because these LTRs in the vector have enhancers, and these enhancers are turning up this LMO2 promoter. All right, so they're not actually acting as promoters, these proviruses, they're just the enhancer parts. So those enhancer parts are gone from the vector. So this doesn't happen anymore. But that's an interesting outcome. So you see, you when you work to treat people, you're going to have these things happen because you're learning. But if you don't take risks, you're never going to learn and you're never going to progress. After that trial, another one was done, I think was just published a year or 2 ago. Gene therapy of infants with SCID with, they were given, again, bone marrow transplants with the wild type gene in a retroviral vector that was modified so it couldn't turn on oncogenes, and all these had functional B and T cells after 18 months. So this can work, and this can save lives. This is really good stuff. A couple more examples. X-linked adrenoleukodystrophy. Here we have a defect in a transporter called ABCD1, and these individuals have fatty acid buildup leading to damage to the myelin sheath of nerves. And so they had patients where they took out their hematopoietic stem cells, infected them with a lentiviral vector with a wild-type gene, put those cells back in patients. So now they have wild-type gene and gene product. This was only a 2-person study, but the demyelination resolved. So here on the right shows the extensive demyelination, and on the left, after the treatment, and their neurologic state is stabilized or improved. So another example of how gene therapy can work. And finally, one more here is blindness. We can make people see again. And these are inherited retinopathies, which are very common, untreatable blinding conditions. And these are monogenic. The patients have mutations in genes in the retinal photoreceptors and the retinal pigment epithelium. So here on the bottom is a diagram of the retina. Here you have your retinal pigment epithelium, and then below it your cones and rods and the nerves that transduce the visual cues up to the brain, allowing you to see. And there are genes in the variety of these cells, and the ganglia, the Muller cells, the rods, the cones, the retinal pigment epithelium that are defective in patients who are blind. And on the right are summaries of various clinical trials to try and restore these genes with different vectors, AAV, lentiviral, and adenovirus vectors. For example, returning the wild-type genes that are defective in the RPE, the cones and rods, the Muller cells, and the ganglia. And many of these have been tested in animal models, and the most promising so far has been AAV. And the way these are administered is they are injected into the retina. The needle is placed right through the eyeball, through the vitreous humor, into retinal pigment epithelium, and then the AAV vector is injected. And because these vectors persist for years, this can restore vision. And so in this one trial for a disease called Leber congenital amaurosis, these patients have mutations in the retinal pigment epithelium 65 gene, which is needed for photoreceptor function. It's defective, so they can't see. In dogs, a single subretinal injection of AAV with the dog gene, and these dogs have the blindness, restores visual function. So after these preclinical studies, it went into human. Eventually, the FDA approved it in December of 2017 to treat blindness. It's called Luxturna. And I interviewed one of the principals who was working on this at UPenn, Catherine High, and you can see that on TWIV 350. So we've had a number of viral gene therapy successes. SCID, I've shown you. ADA, I haven't shown you. The blindness. But by the way, it's costs almost $1 million for 2 eyes. So I guess to see again, it's worth it, but it's a lot of money. You know, I've seen videos of kids who've gotten this therapy and they see their mother for the first time. So I suppose it's worth it. Hemophilia, beta thalassemia, lipoprotein lipase. And now here is the most expensive drug ever, Vexis. It's an AAV9 adeno-associated virus carrying a gene called spinal motor neuron 1 for treating a disease called biallelic spinal muscular atrophy. $2.125 million for this treatment, the most expensive drug ever. But I understand the alternative is a $350,000 drug bill a year for these patients. So I suppose in the long run, this is better. All right, let's finish with viral oncotherapy, killing tumors with viruses. So we want to destroy the tumors. Some viruses selectively replicate in human tumors, and these are animal viruses that people have just checked, and they selectively replicate in tumors. It's really interesting. And then more likely, we modify viruses to target and kill tumors, and these often include immune enhancement. They attract immune cells. And here's a video I made with Linda Coughlin, who did some work with cancer-killing viruses. So this field begins a long time ago in the 40s and 50s. Early studies of human viruses to treat cancers. This is before clinical trial standardization. Hodgkin's lymphoma, they treated it with hepatitis virus. And they didn't even know which virus was in here. This is serum from individuals with infectious or serum hepatitis. 22 patients, 14 of them developed hepatitis and 1 death. Various cancers treating with West Nile virus. Severe encephalitis in 10, mild to severe. This is why West Nile is a BSL-3 agent, by the way, because of this. It can cause encephalitis, but it's mosquito transmitted. There's no reason. Leukemia treated with EBV. These people developed mono. Adenovirus for cervical carcinoma. These are all very poorly controlled. Actually, none of these trials would now be considered capable of yielding any useful information. There are no controls. And within a single investigation, the source of the virus, the route of administration could vary from patient to patient. Concentration was not known and likely differed among patients, the virus particle concentration, of course. And the designs would be totally unacceptable today. For example, delivering to patients serosaliva or tissue extracts from patients that have hepatitis or mumps, the collection of samples from individuals with forms of hepatitis that we know are caused by different viruses, recruitment of patients, even following virus-associated death. Someone dies and they keep going. Now, you may seem outraged that you could treat patients this way. However, many states today and recently, the U.S. Congress have passed, quote, right to try legislation that gives terminally ill patients access to experimental therapies that have completed only phase 1 clinical trials. Nowadays, we're much better. First of all, we know a lot about the properties of cancer cells, and we select viruses that exploit those. For example, here on the left are common properties of cancer cells, which we call cancer hallmarks. Immortality, sustained growth, resistance to tumor suppressors. Okay, these are all features of cancers. And here are the viruses that we have selected to treat various cancers based on these features, like adenovirus for immortal cells, vaccinia for tumors that support angiogenesis, and so forth. Sometimes these are wild-type viruses. Sometimes they are modified to reproduce more efficiently in cancer cells with particular hallmarks. All right, so we say, okay, this cell is invasive or metastatic. How do we design the virus to particularly infect that? So we know this because of careful study of the properties of the cancers. It turns out that fully half of the repressive epigenetic changes that happen when cells are immortalized affect interferon pathways. And so many cells are simply resistant to interferon. And if you look at the data of the tumor genome, this is thousands of patients' tumors, their genomes are sequenced. In one study, of the 2,000 cancer genomes that have been looked at, many of them encode mutations in JAK1, which is a transducer of type 1 interferon signals. So these mutations in the interferon pathway, of course, they let the cancer cells survive immune defenses, but they also make them vulnerable to virus infection, right? So because these cells don't have good interferon defenses, they're susceptible to viruses, and that's really what we're exploiting here. And so we often introduce mutations in viral genes that counter the interferon defenses into oncolytic viruses to limit their reproduction in normal cells. So if you have an interferon antagonist in your vector, you take it out so it won't antagonize interferon in normal cells, but it will replicate in cancer cells because they don't have a good interferon response. So here are just examples of such mutations that impair countermeasures. You want to mutate these so that they will only replicate in tumor cells and not normal cells, right? Because if these viral genes can antagonize interferon, they would replicate in normal cells, and you don't want to do that. Another approach to making viruses specific for tumors, what we call tumor targeting, we can target receptors. Tumors have what we call neoantigens. They have proteins that are not found on other cells because the tumor genome is mutated, right? It's highly mutated because the cells have been growing and mutating. So you have brand new proteins, very different from existing ones, and you can identify those and target viruses to attach to them. For example, you can alter the Measles HA to target tumor markers. One of the glycoproteins of herpes simplex can be engineered to recognize proteins that are abundant on tumor cells. For adenoviruses, you can insert domains that recognize tumor antigen into the fiber, so it will just bind to tumor cells. You can add hexon interlacing proteins that have the same effect, or even put adapters that bind the fiber and retarget it to another protein, so you don't have to modify the fiber itself. So there are many ways that we can have a virus bind to something specific to a tumor cell, which, of course, the important thing there is to find it first. But then once you do, you can make a virus that will target it. Two other things you can do fall under the umbrella of what we call post-entry targeting. So, for example, you can make a vector. You want to kill tumors with this virus. You use a promoter that doesn't work in non-tumor cells. You find a promoter that will only work in a tumor cell. And you have to find that experimentally, but it's possible. There are promoters that only work in tumor cells. And therefore, that restricts your virus to replicating and killing tumor cells. You want to increase the safety, right? So that's positive targeting. You can have negative targeting. Let's say you put into the viral DNA or RNA a sequence complementary to a microRNA. Then in cells with that microRNA, the vector will be inactivated. But if the microRNA is missing in tumors, then the vector will reproduce and kill the tumor. So what you have to do is you have to screen cells and tumors for microRNAs. And here's a northern blot for 2 microRNAs, miR124A, LET7A. And so you can see here we have one that is present only in brain and spinal cord. And this one is ubiquitous. The key would be, of course, to find ones that are everywhere except in your tumor, because in the cells where they're present, non-tumor cells, they'll inactivate the vector. So that's post-entry targeting. Lots of tricks that you can do like this. Another one is to arm your viral vector with a prodrug convertase. So remember, acyclovir is a drug to treat herpes simplex virus. When the drug gets into cells, it's phosphorylated by the viral TK and then triphosphorylated by cell TKs, cellular kinases, and eventually gets incorporated as a chain terminator by the polymerase. Now, if you make a tumor vector and incorporate the TK gene into it, now you could also give the patients a drug that will be phosphorylated by the TK. And that will not only kill the cell, but maybe surrounding cells in the tumor as the drug diffuses out. So we call this arming vectors. TK is one of a class called a prodrug convertase, right? It's a prodrug. It has to be converted to an active drug. We can also put in ion transport proteins, as you'll see in a bit, and immunostimulatory factors. We can clone in the genome of these viruses proteins that will attract immune cells to help destroy the tumor. Another virus being exploited for tumor therapy, Myxoma, the same virus introduced in Australia to kill rabbits. It doesn't replicate in humans, and it turns out to infect many types of human cancer cells. Again, because they have poor interferon responses and they're transformed, which, if you go back to that table, some of those make it more hospitable for virus reproduction. And a whole variety of tumors are being explored here. These are some experiments where we have different kinds of cancers where we transplant them into mice. And so in some cases, these are human cancers. We treat them with Myxoma, and then we put them in mice to see if the tumor cells are killed as opposed to the non-tumor cells. And you can see often we have mice free of multiple myeloma cells in the bone marrow, for example. So sometimes you want to give a patient lethal chemotherapy to kill all the tumors. That's also going to kill the bone marrow. So you take out the bone marrow and preserve it pre-chemotherapy. You don't want to put that bone marrow back in the patient unless you can assure that it's free of cancer cells. And one way may be to treat it with a virus like Myxoma virus. And that's what these experiments are actually trying to do. Measles virus is another one used for killing tumors. In particular, the vaccine strain preferentially replicates in tumors because the mutations selected for that make it a vaccine turn out to make it unable to antagonize STAT1 and MDA5 innate sensor. And so this preferentially grows well in tumor cells. And what they've done is they put a symporter gene in here, the sodium iodide symporter. So now you have this virus producing the symporter in a tumor cell. You then give them a gamma-emitting isotope, which helps to see which cells are getting the vector, because the gamma-emitting isotope will go through the symporter into the cell. And then you can also give beta-emitting isotopes, which will go through the symporter and kill the cell as well. So the symporter is a way of visualizing the tumor and killing it. This has been used in treating multiple myeloma. Multiple myeloma is a tumor of plasmacytomas, of the cells that make antibodies. And normally, these are FACS scans to show you that normally your plasmacytoma cells are making all different antibodies, so they're not clonal, normal PCs. And so here's one patient you see has normal PCs, but also clonal PCs, which is what multiple myeloma partly is. It's a tumor of these plasmacytoma cells. They're multiplying uncontrollably. There are also physical tumors in these patients. And this two patients were treated with intravenous measles virus. And here, post-measles treatment, no more clonal PCs left. So you've killed all the tumor cells in patient 2 who had no normal PCs. After treatment, no more clonal PCs, and the normal ones were returning. And one of these patients had complete remission. And these are brain scans of one of these patients. You can see pre-treatment had a mass on the skull here. It's a myeloma. And post-treatment, it's gone. So the virus got in and destroyed it. So this is being explored as well. There is actually a licensed FDA-approved herpes virus, Imlygic, where the herpes virus is modified in a variety of ways. It has a gene for GM-CSF, which stimulates the production of granulocytes and macrophages. It attracts them. So here's the tumor cell infected with herpes, making GM-CSF. It's attracting immune cells to help kill the tumor. It has viral deletions that cause tumor-specific replication. Then ICP47 is deleted, so there's no inhibition of antigen presentation. So these cells could be killed by CTLs. And after a phase 3, this was licensed in 2015. A couple more just to give you an idea of the breadth of this. Vaccinia virus, JX594. This is a company called Generex, named after Edward Jenner. And these vaccinias have GM-CSF. The TK is deleted, and this is for targeting tumors that have elevated TK. And here they want to know, if we put this virus intravenously, will it reach metastatic tumors? Because you can't always inject them into tumors. So they treated a number of patients with advanced tumors of various sorts, treatment refractory. They didn't respond to anything else. And in half the patients, the virus replicated in tumors after IV inoculation, and they had activity in half of the patients. So in these stains, you can see there's virus in cancer cells, but not normal cells, rectal cancers, endometrial cancers, and colon cancers. So the virus is reaching these cells specifically. So it's a proof of concept that you can deliver virus intravenously and get it to the cells that you want. And another example of prodrug convertases. Remember, TK converts prodrugs to inhibitory drugs, ganciclovir to ganciclovir triphosphate. You could put cytosine deaminase in, which converts 5-fluorocytosine to 5-FU. And these two, the triphosphates, stop DNA replication of tumor cells. You deliver them via the virus, you get them specifically into tumor cells. Drug is converted from the prodrug into the toxic metabolite, which will kill the tumor cell and will also kill bystander cells, and that's good if it's a big tumor mass. And that's an example of this particular virus, TOCA511, which is an amphitropic murine leukemia retrovirus with cytosine deaminase. So the virus is either injected into the tumor or it's injected into the tumor. So here we have a brain tumor. The virus is injected in. Then they give the patients intravenously 5-fluorocytosine, FC, which then in the tumor only where the virus is converts it to 5-FU, which kills the cell. So that's the basis for that. And it's in phase 1 and 2 for gliomas. A modified polio is being considered for treatment of glioblastoma. And this is a poliovirus Sabin, a vaccine strain with the internal ribosome entry site from a rhinovirus, which makes it attenuating, not paralytic. These tumors upregulate the polio receptor. So this has gone into phase 2 for glioma and has increased the median survival time. So it's an interesting way of repurposing a vaccine strain. And finally, one last example, reoviruses, one of the many viruses we've talked about. Unmodified is not pathogenic for humans. It was found many years ago to kill cells with an activated RAS pathway. RAS, again, one of those oncogenes in that mitogenic pathway picked up by a retrovirus. And those cells with activated RAS are particularly susceptible to killing by reovirus. And this has been tested for head and neck and many other tumors. And here's the company, Oncolytics Biotech. And I put it up here just to show you this slogan, innately adaptive. So that in the end, the therapeutic efficacy of these viruses turns out to be a combination of the virus killing the cancer cell, but also activating anti-tumor immune responses. So the virus, infects the cancer cell, it kills it, but the dying cell releases mediators that attract immune cells of various sorts, which will then recognize and kill the tumor cell as well. So you're doing a general activation of the immune response, which has been quiescent in the face of the tumor. That's what the virus is doing besides killing the tumor. And in many cases, it will turn out that virus killing of the tumor is probably a minor part of elimination of the tumor. And that brings us to the last therapy I want to talk about, and that is cancer immunotherapy using CAR T cells, which was approved by the FDA in 2017. And it's all based on having an antigen identified that's unique to a tumor cell. And then you build a chimeric antigen receptor that recognizes it. You first make a single-chain antibody that recognizes this tumor antigen. It's right there, the targeting element. It's a single-chain antibody specific for this tumor antigen. You then make a chimeric antigen receptor with that single-chain antibody, transmembrane domain, and then co-stimulatory and signaling domains so that when that CAR protein is present in the T cell, it will interact with the tumor antigen and the T cell will kill the tumor cell. So the way this is done, one iteration, of course, is you harvest the T cells from the patient. You deliver this gene via lentivector. You have to, of course, first find out if the tumor antigen is one of the ones to which you have a single-chain antibody. You put it in a lentiviral vector. You deliver it to the T cells. Then you put the T cells back, and they will kill the tumor cells. And this has been very effective. One way of targeting tumor cells by immunotherapy. It certainly will be combined with viral oncotherapy as well. So this is an amazing expanding field, and it's all using viruses. None of this would be possible without viruses. And that leads me to end by telling you the importance of basic research. These therapeutic viruses, they have all been made possible because scientists are making fundamental advances in virology, recombinant DNA, immunology, and clinical science. And a lot of it is by scientists just being curious without knowing what in the end is going to come out of it. And we need to have that. There has to always be a balance between basic research, letting scientists be curious, and translational research where we apply what we learn in the lab to patients. There's a lot of push to do more and more translational research, which is focused. I'm going to cure diabetes. I'm going to cure cancer. That's not always the way it works. It often works by an accidental finding so that you can develop a vector to kill a tumor, for example. So we have to balance this, and that's what I want to leave you with at the end of this course, because we always have to have basic scientists being curious and clinicians to apply the curiosity. I want to thank you for taking this course. This is day one, way back when we were in a classroom. And in that first lecture, I talked about the spreading virus, which ended up canceling our in-person class. Amazing that it happened. But here we are at the end. A few more administrative things. There is a survey at CourseWorks. Please fill it out. Let us know what you think of the course. Finish the quizzes. There was one more put up yesterday. That'll be the last one. I will have office hours on Thursday from 4 to 6 p.m. And if you can't make it, you can email me questions. We can set up other times to chat. I'm happy to answer your questions. But really, most of all, don't forget what you've learned here. I told you in the beginning, if you want to understand human health and disease, you have to understand viruses. I've given you tools to do that. You now know more virology than 99% of the people in the world. I think you have all the tools to understand what has happened so far with SARS-CoV-2 and what's going to happen in the next few years. So don't lose it. It's really good information. And you can still learn from me and be curious because I'm all over the place, as you know. This, by the way, is my license plate because I love viruses, but I have a whole load of podcasts and a blog. Follow me somewhere, and I will continue to teach you virology, Twitter, Instagram, YouTube, Facebook. There, all these places I use to teach about viruses. And follow me and learn more and more. So this doesn't have to be the last time you hear from me. So thanks once again, and have a great end of semester. All right, everybody. I'll miss you. Bye.